Our next talk is going to shed light on us because it is about using fluorescence to get diagnosis information. And it's going to be presented by Eva Galvez, who's a chief scientist at the Instituto of Carboquímica of Consejo Superior de Investigaciones Científicas, where she leads a group of fluorescence-based immuno and nanobiosensors. She has many published papers, and she's co-inventor of a couple of patent licensed to companies, and she runs several projects national and internationally. Her research interest focuses on the development of biosensors and other analytical techniques applied to the detection of biologically unique molecules, but also on the diagnosis of immune system-based pathologies.
Okay, thank you very much. And I would like also to thank the organization for giving us the opportunity to present our main research lines related to immune biosensors for cancer and autoimmune or inflammatory disease. One of our main research lines are the development of analytical biosensors based on fluorescence resonance energy transfer. Very quickly, fluorescence energy FRET is a dependent distance energy transfer from an excited donor fluorophore to a suitable acceptor fluorophore, as we can observe here. In this way, these FRET-based biosensors allow us to monitor protein-protein interactions and protein dynamics, as we will see later. We applied these biosensors to monitor the immune response, but for what? Good, to look for biomarkers to analyze the efficacy of cancer immunotherapy, and also to look for biomarkers related to inflammation and or autoimmune disease. Now I would like to show you two types of biosensors that we are currently developing to monitor key steps of the immune response during cancer and autoimmunity. One of them is related to the analysis of cell-cell interactions mediated by a family of proteins named integrins. And the other one is Gramsame A, a new inflammatory molecule that is related to the generation of the inflammatory response that induces tissue damage during autoimmune disease. It seems quite complicated, but it's just an example of the implication of the integrins in the immune system. Now I'm going to show you two examples of two biosensors we have developed. One of them is these biosensors to monitor protein-protein interaction. As we can see here, when two proteins that bond to two different fluorophores are interacting, we can see a FRET-on signal. But when this interaction disappears, the fluorescence emission is lost since there is not anymore energy transfer. We have applied this biosensor to analyze the interaction between the integrin LFA1 molecule and the adhesion molecule ICANN1, since they are involved in several processes as cell cytotoxicity, cell migration, and metastasis, among others. That could be useful to test some drugs for the treatment of these pathologies in which this interaction is involved. The other example I would like to show you is this FRET biosensor to monitor protease activity. In this case, we are able to monitor if a protease is able to cleave a specific peptide substrate functionalized with a donor fluorophore and a quencher. In this way, if the protease cleaves the peptide, the quencher is removed and we are able to see the fluorescence emission from the donor fluorophore. With this type of sensor, we want to monitor granzyme A activity, because it is related with the development of inflammatory colorectal cancer. In order to apply this biosensor for early diagnosis and prognosis of this type of cancer. Here is the people involved in all this work, and thank you very much for your attention.
Thank you, Eva.